Dear student, in this video we will learn about how the sound wave is converted to an electrical wave and how it is transmitted to the brain. So we know that the sound is actually transmitted first via the external ear. So what are the parts of the external ear? It is first part is the auricle or pinna. We can see here the pinna is a flap of elastic cartilage. It is covered by skin and it is fixed in case of human being. And what is the function of this pinna? It funnels the sound wave and helps to establish the direction of the sound wave. What are the parts of the pinna? You can see here the auricular part, the outer part, it is known as the helix. Inside the helix we have the antihelix, and there is a triangular cartilage near the external auditory canal or meatus, that is known as tragus, and the lower part, lower soft tissue portion, is known as the lobule. Okay, and at the just above the lobule we have the antitragus, just the another triangular elevation. Now the sound will enter into the external auditory meatus. So this is the external auditory meatus. It is a tube like structure and it conducts the sound wave to the middle ear. External 1/3 of this tube, it is cartilaginous, and the internal 2/3, it is bony. So one point is important here: when the sound is transmitted through the external ear to the middle ear, there is usually no modification or distortion of the sound, but there may be distraction if there is impaction of wax or hardening of wax. How the wax is secreted? There are some ceruminous glands present in this external auditory meatus. These are the sebaceous glands. They secrete the wax. If there is impaction, then there will be some conduction defect in the external ear. So there will be rise of 15 decibel or increase in the sound intensity, that is done by the external canal and the pinna together, due to their characteristic shape. Now when the sound it is coming to the ear, so it will cause the vibration in the eardrum or the tympanic membrane, which is the entry point of the middle ear. So now we will look at the middle ear cavity. It is just like a matchbox cavity which has got 6 walls, as anterior, posterior, superior, inferior, medial and lateral. We have here the lateral wall that is formed by the tympanic membrane. We have removed the tympanic membrane. Now we are seeing inside the wall. Now the anterior wall, we can see here, it opens into the exterior via the eustachian tube. The posterior wall, it communicates with the mastoid air cells via the tympanic antrum. The mastoid air cells, there may be inflammation and that will cause the mastoiditis. The medial wall, it consists of two small membranes. You can see here, one is the oval window, another is the round window. Middle ear is actually connecting it, this windows connecting the middle ear to the membranous labyrinth of the inner ear. Now the lateral wall is formed by the tympanic membrane, as I told you, and the roof of the middle ear, it is formed by the tegmen tympani that separates it from the middle cranial fossa, and the floor is formed by the actually it separates it from the jugular fossa. Okay, so the lateral wall, as I told you, is formed by the tympanic membrane. Now what is the structure of the tympanic membrane? It is a thin elastic membrane. Its surface area is 50 to 90 millimeters square, and its apex with its apex towards the middle ear. We can see the handle of the malleus, which is a ossicle, which is actually attached here, and the tympanic membrane at the inner surface, it is covered by mucous membrane, and the outer surface, it is covered by the skin. Inflammation of the tympanic membrane is known as the tympanitis. Now what is the function of tympanic membrane? It acts as a resonator, so helps in the transmission of vibration, and it also acts as a mechanical barrier that prevents the entry of foreign particles, dust and other things from the outer ear to the middle ear. Now come to the inside the middle ear. Inside the middle ear we have the three ear ossicles: malleus, hammer, which is also known as hammer, incus and the stapes. The handle of the malleus is attached to the tympanic membrane. Here we can see here, and the malleus is bound to the incus by minute ligaments. So whenever malleus moves, so incus moves with it. Okay, now come to the stapes, the purple bone. The opposite end of the incus, it articulates with the stem of the stapes, and the footplate of the stapes is attached with the oval window, which is the membranous labyrinth. Okay, so in this way they are attached here. Now we will see the attachment of the ligaments and the muscle. Two important muscles are attached over here. One is the tensor tympani muscle, which pulls the handle of the malleus inwards, thereby it increases actually the tensions of the tympanic membrane. This makes the membrane more responsive to the sound waves. Okay, so this is the function of the tensor tympani. Another muscle is there, that is the stapedius, which is the smallest skeletal muscle in the body. What is the function of it? It helps in response to loud noise. It contracts and pulls the footplate of the stapes out of the oval window. So it dampens the vibration, prevents the ear from damaged from the loud noise. Okay, so if there is paralysis of the stapedius muscle, so the patient will be abnormally sensitive to loud noise, that is known as hyperacusis. And the diseases associated with the movement of the ossicles, if the movement it becomes very less and they becomes more stiff, then that is known as otosclerosis, and that will result in a profound conductive type of deafness. Okay, so now this contraction of this stapedius muscle, and to reduce the intensity, this reflex is known as the tympanic reflex, or also it is called the acoustic reflex, which is a protective reflex. Now, what are the functions of middle ear? It receives the sound wave, it transmits the sound wave, it helps in the equalization of air pressure. How? Because it is connected with the eustachian tube, so that helps in the equalization of the air pressure. It protects against the prolonged loud sound. And last important point, and that is the most important point, it helps in the amplification of the sound waves. Why this amplification is required? Because it is required for the impedance matching. What is impedance matching? So sound wave, we know that it is when it is passing through the external ear and the middle ear, it is passing through the air. Now when it will enter into the cochlea or membranous labyrinth, it has to vibrate the fluid. The fluid has the greater inertia. Okay, so we need more force to cause the vibration of the fluid. If it is not increased, this force is not increased, so what will happen? The sound will be imperceptible by the brain. So there must be a matching between the sound wave air vibration and the sound vibration of the fluid. This matching is actually done by this known as impedance matching, and it is done by this amplification. How this amplification actually happens, we will see here. There will be how much amplification it happens? 22 times amplification. First amplification, it is done by the tympanic membrane, the difference of surface area of tympanic membrane and the oval window. The surface area of tympanic membrane is 50 millimeter square, and that of the oval window is 3 millimeter square. So the ratio is around 17:1. So the total force of sound wave, it is exerted on the tympanic membrane, is transmitted to the oval window. But as the surface area is decreased, so the force per unit area, that is the pressure, it will be increased by 17 times. Okay, so this is 17 times increase. Another is ossicular chain will act as a lever. Okay, so that while transmitting the vibration, because the handle of the malleus is 1.3 times longer than the long process of the incus. So handle of malleus is longer than the long process of incus. How many times longer? 1.3 times. So there will be enhancement of force of 1.3 times. So 17 times by the change of surface area, and 1.3 times by this lever action. So 17 into 1.3, that is around 22 times amplification will be done, and it actually performs the job of impedance matching. Now come to the internal ear. So internal ear, you can see it is a cochlea. Cochlea has two parts. One is the bony cochlea and another is the membranous cochlea. Okay, in the membranous cochlea, actually there will be actually this transducer action, that is conversion of sound wave to the electrical wave, it will happen. Now, And this membranous labyrinth has got three tube-like structure. You can see here, one is the above is the scala vestibuli, middle is the scala media, lower part is the scala tympani, and they are separated by two membranes. Scala vestibuli and media is separated by Risner's membrane, and scala media and scala tympani is separated by the basilar membrane, and the fluid present within them, the scala vestibuli and scala tympani, they contains the perilymph, whereas scala media, it contains the endolymph. There is constitutional difference between these two fluid. Okay, another important point is that both the basilar membrane and the Risner's membrane, they actually coincides or fuses at the apical part. There will be a narrow gap between at the apex between this membrane and the bony labyrinth, that is known as the helicotrema. Okay, so here there is a speciality of the Risner's membrane too. This Risner's membrane, it is so thin, so it's easily moved so easily moved that it does not obstruct the passage of sound vibrations from the scala vestibuli into the scala media. So as far as the fluid conduction of sound is concerned, this vestibuli and media, it will be considered as a single chamber. Now this scala media contains a specialized fluid which is known as the endolymph, okay, that is secreted by the stria vascularis. Now the stria vascularis actually has got huge concentration of sodium potassium ATPase pump. So what it does is stria vascularis pumps potassium into this fluid and so takes out sodium from it. So endolymph is very rich in potassium. That is a speciality of endolymph. So here we can see the scala vestibuli, scala media, scala tympani, and the helicotrema by which they are joined. Another important point is there, this scala tympani, it is connected to the middle ear by another window. Above we have seen that footplate of stapes is connected to the middle ear to the internal ear via the oval window. Whereas here this scala tympani, it is connected to the middle ear by another window which is known as the round window, and this round window is covered by a flexible membrane which is known as the secondary tympanic membrane. Okay, and the direction of movement of this oval window and round window will be in opposite side. Now we will see how this conversion of sound wave to the electrical wave it happens. It actually happens by a specialized organ present on the basilar membrane which is known as the organ of Corti. This organ of Corti, we can see here lies actually the receptors for the hearing. For example, the photoreceptors or for the vision receptors are rods and cones. Just like that, the hearing we have the receptor, these are the hair cells. There are two groups of hair cells we can see. Okay, this organ of Corti is actually the transducer which convert the mechanical energy to the action potential of the cochlear nerve. Two group of hair cell we can see. One is the on the outer hair cells and the inner hair cells. There are and they are separated by the rods of Corti. The triangular gap between the rods of Corti is known as the tunnel of Corti. The inner hair cell, it is the lesser in number, 3500. They form a single row and extend along the entire length of the cochlea, whereas on the lateral to the tunnel of Corti you can see the outer hair cell. They are 20,000 in number and they are arranged in 3 rows. Okay, in addition to that, there are some supporting or sustentacular cells as well. Now all these cells, they have the cilia above them, and the cilia of the hair cells, they touches a gelatinous tectorial membrane that covers the organ of Corti which is present, and this viscous membrane contains collagen and glycoprotein, and it is attached to the medial edge. Now what is the speciality of the hair cell? Outer and inner hair cell, their structure are almost similar, but the outer cell is a bit longer, and the ciliary part of the outer hair cell, it touches the tectorial membrane, whereas in case of inner hair cell they do not. Okay, another thing is that these cilias, they are actually the stereocilias. Okay, now the stereocilias, they have a speciality. We can see in the next pictures, these stereocilias, they are connected with each other by tip links. What are those tip links? Tip links are mechanically sensitive cation channels or transduction channels. Okay, they are present at the junction of the cilia, and these tip links help the bundle of the stereocilia to move as a unit. Okay, now the stereocilia, you can see their height gradually increases towards the lateral edge of the ear cells. Therefore, the row of stereocilia towards the stria vascularis, it is the tallest. We can see here towards the stria vascularis, they are the tallest. Okay, these stereocilias. Now, how actually the electrical signal is generated? When the shorter stereocilia, so this vibration is there. Okay, when the basilar membrane is vibrating, so what happens, the shorter stereocilia, it bends towards the taller stereocilia, okay, towards the stria vascularis, and the cation channels, these tip links, they open, and when they open, they allow the entry of potassium. Okay, so potassium will enter into the cells. So potassium positively charged ion, so it will cause the depolarization, which will cause the opening of voltage gated cytoplasmic calcium channel. So calcium, voltage gated calcium channel, so calcium will enter into the cell, cytoplasmic calcium concentration will increase, and it will help in the release of the neurotransmitter, that is the glutamate, at the base of the synaptic cleft. Okay, so first what happens, that is just shorter stereocilia bending towards the taller one. So tip links are opening, potassium is entering, depolarization, opening of voltage gated calcium channel, and release of neurotransmitter glutamate. Okay, that will cause the generation of action potential in the cochlear nerve. Okay, so now the question arises, why the potassium is entering? Because this, the answer lies here. The, we can see, if we look at the hair cells, okay, the resting membrane potential of the hair cells, that is the endocochlear potentials. Okay, we can see that the hair cells, they have two surfaces. One is the basolateral surface, another is the apical surface. Okay, the basolateral surface, the basolateral part, it is actually potential, it is around -60. It is due to the continuous efflux of potassium across its basolateral membrane. Okay, so because this basolateral hair cell, basolateral surface is bathed in the perilymph, you can see in the scalar tympani, it is bathed into the perilymph, which has low potassium concentration, whereas apical surface of the hair cell, which contains the stereocilia, it is bathed in the endolymph, which has higher potassium concentration. So relative potential is +80. Here you can see it is above, it is +80, below it is written -40, it is actually -60. So if we see the potential difference, then 80 millivolt minus -60 millivolt, that is 80 plus 60, around 140 millivolt. Okay, so a large electrical gradient is there that favors the cation influx, especially the potassium, through these transduction channels, and that's why there will be depolarization and generation of action potential. Okay, so action potential is generated. Now into the cochlear nerve, in the from it will go to the cochlear nerve, through then it will go to the cochlear nuclei. From the cochlear nuclei, it will cross to the opposite side and it will go to the superior olivary nucleus. From the superior olivary nucleus, it will form the lateral lemniscus, and it will go to the inferior colliculus. In the inferior colliculus, from that it will enter the next order neuron. It will go to the medial geniculate body, which is the part of the thalamus, and from there the last order neuron, it will go to the primary auditory area number 41 in the brain. So in this way we understand how actually the sound wave, it is transmitted from the ear, then into the internal ear, and then the electrical signal and into the brain. Thank you.